He is in UPenn. Dr. Murthy has completed his PhD from IIT Kharagpur in molecular virology in 2007. He has also done 3 postdoctoral fellowships from University of Michigan and Ann Arbor, University of California, San Diego, and University of Pennsylvania. He has also been awarded RO Genentech Research Award in 2012 and Helen Jonetta Reed Memorial Award, USA. So RO is the biggest research, it's called the Association of Research and Vision and Ophthalmology, which is the greatest research right where people like you or from Chemical Technology Institute or even a Vivekananda Engineering, any faculty which is working in anything to do with the basic science of eye and for vision, they can take part. So that's the only field where the doctors will be less and Nobel laureates come and give orations. And he has also been, in addition to that, Dr. Murthy is also a member of the National International Professional and Scientific Society. He initially became a part of the SUR Review Committee, Association for Research and Vision of the SARU in 2006. Currently, he is a Developing Countries Eye Research Fellowship mentor. And he is an academic and institutional committee member in MPhil. He is the external examiner for Dr. CNG UK. And he has given guest lectures in the model systems to study the role of epigenetics and glaucoma at Harvard. And he was a co-organizer of Stem Cell RTC Special Interest Group. He has done research publication in bioinformatics to an approach to primary open angle. He has published several papers, including the silencing the expression of CTRP5, C1, 2PNFS, and ELO, for genes by small RNA in a book named Advances in Experimental Medicine and Biology. And so when you open it, so the idea of back to India, you have a good lab. Because I think the UPenn lab, I've been there to the lab, and they are doing wonderful research. And the chairman is she's got 11.5 million grant for doing research on African American glaucoma. And he's part of that. Plus, now he told me today's talk will be induced pluripotent stem therapy for treatment of retinal ganglion cell death in glaucoma. Everybody keeps asking if they're not a treatment for glaucoma because either you put drops, which I do twice a day, or you have to operate. And still, with the risk of it's a slow, silent vision killer. So we want to work on several things, and that's our approach for Pratiksha. And we have Runali with us. I know some of you here. And we have, I got Dr. Anjesha and Arsha. They are running a pharmaceutical industry, a pharmaceutical factory in we also have research time in Ahmedabad. And so I hope they will be interested in doing something with this once we establish something. And we should do something so that Dr. Murthy like person can come here. And already there are a lot of PhD people working. Thank you very much for in a short time you give us this place. Thank you. Over to you. Can I finish? No, I'm good. Yeah, already.
Thank you, Dr. Nasarjan. Such a kind introduction. Always a pleasure to come by. This is my second time visiting Mumbai, and Dr. Nasarjan's hospital. So I'm an assistant professor in the Department of Ophthalmology in the University of Pennsylvania. Am I audible to you in the back? Yes. I can speak more loud, or maybe I need to get the mask off or something. Please. Is it clear now? Yes. Oh, thanks. So this is one of the projects that we're doing in the lab. I work on, as Dr. Murthy mentioned, I did 3 postdoctoral fellowships at 2 different places. And my initial postdoc, I did my PhD in IIT Kharagpur, and the topic of my research was molecular biology. And the core concepts that I learned in molecular biology was from there. And I did my postdoctoral fellowship. I applied those concepts. I took along with me, I learned, and then applied it to ophthalmology. And ophthalmology was my big time fascination. As you all know, there's a saying, eye is a window to the brain. And, you know, I always wanted to study brain, and eye is part of it. And then, you know, I pretty much got excited with the initial offer I got from University of Michigan. So I initially pursued my research on retinal degenerations. And I worked there for like 4 years before I moved on to my second postdoc, where my research got much more broader from a single monogenic diseases to a complex disease like AMD, complex disease like AMD. Mostly got interested in the genetics of AMD initially, then the functional aspects of AMD. Then you study the function of genes which are associated with one particular disease or a complex disease, because it's not a monogenic inheritance anymore. Then I switched gears, and when I came back to UPenn, I continued pursuing research on AMD, but I also collaborated with the chairman of ophthalmology, who was working on a glaucoma project. And I'm sure you already know that there have been, I wouldn't say therapies, but there were some interventions by using anti-VEGF drugs for AMD. But for glaucoma, the traditional treatment was using drops. The layman's understanding of glaucoma is to have some DB in the eye or pressure in the eye, and the only way you solve it or you only treat it is by putting a drop. And you put a drop today, you may forget tomorrow, and you come back again for another day, and then people and the patients do not follow. And that's why glaucoma, even though one, it is not detected early, two, even though it's detected, it's not managed well enough. So obviously we have to come up with better treatment strategies, and one of the aspects I was looking into is to see if there is a cell-based therapeutic approach that we can explore, which mainly looks at the core problem in the glaucoma and just not tackling the IOP. I'll tell you more about what IOP is, the anatomy of the eye, and everything. But this would be a hardcore vision research kind of topic. So stop me anywhere you have questions on, and then I will be happy to brief it. So yeah, the topic of my talk is to use pluripotent stem cell therapy for treating RGC death. So why RGC death and what's glaucoma? Let's briefly look into it. Okay, before I just want to put it out there, I have no financial disclosures yet, but the work that I'm going to talk today, we have filed a professional USA patent for the cells that I used in this methodology. So it's still waiting for a company to pick it up. We are in the place of CDAs, doing CDAs now, but it's not picked up completely yet. So there's still some time before we move ahead with the paper. So glaucoma is an optic neurodegenerative disease. It's characterized by structural and functional deterioration of the optic nerve. Optic nerve is the nerve that connects the eye to the brain, and it's a leading cause of blindness in the world. And glaucoma there are several kinds of glaucoma, but there are two major types. One is the open angle glaucoma, and the second one is the angle closure glaucoma. And in India, it's approximated that there are 11.2 million individuals over the age of 40 who have glaucoma. And among them, 6.48 million have open angle glaucoma. So primary open angle glaucoma is definitely one of the major diseases here. And I want to show you a picture. So this is an image how a normal person sees this particular image. And this is a patient with glaucoma. If you see the same image, this is how it looks to him. So they have a loss of peripheral vision. I'll tell you, peripheral vision is actually, you can see in the center, all the neurons, in the rest of the eye, they fade away. They lose sensitivity to light, and they have problems with night vision, and they also have blurred vision. It's a big introduction, and I'm sure most of you know this. This is the eye, and for the purpose of explanation, I divided. This is the anterior segment of the globe. This is the posterior segment of the globe. In the anterior segments, you have the posterior chamber and the anterior chamber here. So aqueous humor is a liquid that actually gets secreted in the ciliary body, and then it actually goes against the pressure in the eye, and it gets secreted through these two pathways, through the squamous canal, through follicular mesh. So this is a normal eye, and this flow of aqueous humor keeps the eye pressure intact, eye bulge, and eye happy. Once there is an obstruction to these pathways, then there is a pressure build up in the front of the retina, in the front of the eye, the globe, which gets pushed on to, pushed onto the back of the eye, and it mainly affects the cells. Some of the cells here, these are the retinal ganglion cells right here, which are affected by this increased pressure. So I kind of moved it up, and this is the retina where you see these are the photoreceptor segments, and then the light comes through here to the lens, and then it falls here. And this is the retinal layer, it's a multicellular layer. The light hits here, the light stimulus carries all the way back to these ganglion cells. These ganglion cells can take the light stimulus and then connect to the brain through optic nerve. And in glaucoma, due to increased pressure, what happens? This is a normal retina where you have these RGCs. They're all, I call them RGCs, the retinal ganglion cells. They're all connected, and then they dive into the optic cup. And then this is a happy eye. Where in glaucoma's eye, due to increased pressure, the ganglion cells die, resulting in, you see the number of fibers going through, they skin down a little bit, and then they increase the optic disc and the optic cup. So you have a bigger margin here, and this tells you that there are less number of retinal ganglion cells here than compared to the margin. And the patient slowly starts to lose sight of the vision. So two types of glaucoma, as I said, the open angle and closed angle glaucoma. The open angle and closed angle is based on the angle it creates when there is a blockage. And there's a blockage if the angle between the cornea and the iris is large, is open, then it's called open angle glaucoma. But if it's closed right here, it is called closed angle glaucoma. So to understand the pathobiological mechanisms of RGC loss in glaucoma, what we need is to establish pure retinal ganglion cell cultures. Because if you want to study what pathways affect RGC death in glaucoma, you need to have pure cells which can go back and then probe them in vitro, use some chemicals, understand mechanisms. And it's more of a cell-based approach than an animal-based approach, which obviously you are promoting, right? So we wanted to establish a cell culture which mimics the RGCs in a dish. And to do this, there have been some protocols, like they started 3 years ago when people started using embryonic stem cells. And then, after the introduction of Yamanaka factors, using them for stem cell differentiation experiments, people all over the world started using them to actually, tailor-make the cell types they want, and then try to see, put them in a dish, and then kind of, study mechanisms, disease pathways, because that will eliminate the use of animal model systems. So, for actually using the RGCs too, people have tried using embryonic stem cells, and they also use pluripotent stem cells. But they have developed protocols. I'll tell you, they have developed protocols which are not as efficient as they should be. Because what they used were, they used to take initial experiments where they took the human eye, a human donor eye, and then they used MAC sorting, which is a magnetic sorted cell sorter. So they use MAC sorting and an antibody which recognizes retinal ganglion cells, and then they try to pull these retinal ganglion cells together. But the yield of these ganglion cells that you get is like 20%. So it's not really efficacious protocol. And Oh, sorry. And 4 years ago, Zan Lab developed a protocol where they have modified iPSCs with a promoter which is specific for ganglion cells. But they use CRISPR-Cas9 technology, and then they have actually engineered a RGC specific promoter called the BLINDFREE, and then they introduced the GFP in the promoter. And what they did was they developed a screening cell line, which is, I think, very common nowadays to kind of screen the population of cells to identify what they want and selectively pick it up and then amplify it. And by doing that, they have increased the efficiency of making RGCs in a dish by 45%. So it's quite an improvement from the primary cultures where you're just taking human retina cells. And people also have used, so after the success of 3D retinal organoids, people have used dissociation methods, like manual dissociation methods. So they take these organoids, 3D retinal organoids, and they split these cells, all the 9 layers in the retina I showed you before, and then they use max sorting, and then they isolate these cells. That still improved the purification by like up to 45%, but you still have a large chunk of RGCs which you cannot capture. And I did not tell you, but RGCs, there are over 42 types of retinal ganglion cells in the human retina. And not all these isolation methods are able to capture them because the antibody that you use are only select a majority of them, not all of them. So you do have a disadvantage of using the earlier techniques. So we thought, okay, what can we do in this space? I did actually collaborate with Jean Bennett. I'm sure you heard about her. She is a faculty in the University of Pennsylvania. She's the one who pioneered bringing AAV cocktail for RP65 and making a blind person see. So we collaborated with her, developed this technology a little bit, then came up with a unique protocol very specific to our lab. We wanted to see if we can push this barrier and then make sure we actually get more than 90% purity of these RGCs. So we ended up actually developing a highly reproducible, chemically defined two-step protocol. Why I say two-step? So all these iPS RGC differentiations, even though they are done in a dish, they have a method that we need to follow to be able to get the cell type that we want. So they mimic the development that happens in the eye. We have to mimic that because there's no shortcut to making RGC that you want. So you have to follow all the biological mechanisms that happen in our body before these cells are formed in humans. So we recapitulated them. We recapitulated most of these pathways by using some small molecules and some antibody cocktails by blocking some inhibitors along the way. And we actually developed this protocol wherein the iPSCs are initially taken into a retinal progenitor cell state, and the retinal progenitor cell state is eventually differentiated to a ganglion cell state. These retinal progenitor cells are the cells that would be the seed for making all the photoreceptor out segments. So those can be used for treating all the retinal degenerations. And the ganglion cells that we develop after this can be treated for use for treating glaucoma. So it's a two-stage approach, but beneficial at both ends, depending upon where you want to stop the program. So yeah, we inhibit SNAP pathway, and then we also used inhibited root pathways to do this. We published this protocol. So this is, I'll not bore you with the details of this, but I'll tell you, it takes up to 25 days or like up to 36 days to do this in a dish. Compared to all the available techniques out there, this is one of the fastest, the development is pretty fast here. The established technologies we have out there are up to 75 days. where you get at least 45% purity of these RGCs with a lot of variability. But here, the reason why we could actually achieve this so quickly was of this two-step approach where we initially focused on getting 100%, nearly 100% RPCs, and we switched a couple of pathways and incubate them for another few days before we take them to maturation. So from day 26, it's technically RGC maturation where you can see these axons develop, and I'll show some videos to support that. So it's still talking about all the clinicals used. You can actually refer to the manuscript we published if you're interested. But what we also did was, in doing this, we optimized actually each of these inhibitors and then tried each one of them. And then we actually screened the RGCs to get out of this, and then eventually decided that if you do inhibition of both, notch inhibitor, ring inhibitor, then you get the RGCs that we want with just purity. I'll actually show you and convince you that the ones that we make are really good. So these are the cells. These are the iPSCs. This is the RPC differentiation I was talking about. This is where the RPC differentiation stopped, and we slowly switch gears into RPC differentiation. So we characterize them like any process. Garbage in is garbage out. So we need to have the best iPSCs that we could ever actually use to start any differentiation, because all these iPSCs have to be highly pluripotent. The pluripotence should be more than 95% to start the protocol. If you start with anything lesser, the yield of RGCs that you will be getting will be drastically low. So we start with iPSCs. iPSCs, I'm sure you know, these are induced pluripotent stem cells. This can be derived from hair follicles, skin, you name it. And we have a repository of over 50 lines. In the US, we primarily work with Caucasian lines and African American lines, two major populations with diseases with them. So we have a repository of more than 30 lines for each one of them. We characterize them thoroughly and genotype them, and then we have a repository. We usually start with full cultures of iPSCs. We bring them to RPC differentiation. We characterize them using facts. So fact sorting, we have some markers, check10 and PI65. We have to make sure that they're really good. And also to tell you, this is an in vitro methodology, not an in vivo methodology. There is a variability to everything you do. And even the same experiment done by two people may lead to two different results, even at the fourth digit after the dot, really on the fourth digit after the dot. So you do have that variability, even like practical. So even these iPSCs, they do show variability. And even with all the variability, we are able to successfully achieve more than like 85% of the markers that we screen. You would see positives for all the lines to develop. There are some poor lines which never develop, so we don't go ahead with them, but like we did optimize the lines that we work with. So we screen for the RPCs, and this is a step we have come up with which really helped us. So if you know, working with retinal organoids or anything like that, what you need to do is a technician going into the chamber and then picking the organoid by hand. You have to hand pick some of these for taking them to the next stages of differentiation. So that was introducing a lot of variability to this protocol. So what we did was we used a technique called cross-hatching. It's just like streaking horizontally and vertically, kind of split them into small colonies and give them enough seed to kind of go to the next step. So we did cross-hatching there at day 24, and this really helped us take care of the variability. And then we do that routinely, robustly, 25 strokes this way, 25 strokes this way, always consistent, and then we don't have to handle. That's the beauty of this, because we used a combination of small molecules and proteins. And with this technique, I mean, by day 35, they start developing into RGCs. Here you cannot, it's a very high max. I'll show you the cells in a little bit. And we characterize them too. Once you get them, we also characterize them using tax sorting, some of the markers for RGC specific genes, it's the control, SMCT is the gamma synuclein, and RBPMS is the RGC marker. As I said, like for every cell type we differentiate, we have to characterize them. We don't get in research. I don't know how it works in industry. Maybe they have one or two time points. In research, we do it at least 3 different ways to kind of make sure that we are really producing what we want to produce. So we characterize them using IHC markers. So these are SOX2, RAX, AX6, CI67, and CHUX10, and then we make sure that most of the cells we develop are RGCs. And for RGCs, we do all these techniques. We do fact sorting, we do immunolabeling. Am I going slow or something? Okay, that's okay. That's okay. Thank you for stopping by. Yeah, because I mean. Thanks. So for the RGCs, we characterize them, a number of ways to do flow cytometry, immunolabeling, constant expression. These are pretty routine when you actually do it in a lab. I mean, you have to check all the boxes to make sure you are developing the cells that you claim you are developing. And what we see, we actually develop more than 90% pure RGCs. And if you want to really take it up a notch and really, increase the yield, you can see your, max sorting. And here you are defining what you are isolating. But here, if you stop right here, you are developing different kinds of RGCs. So these are the different markers and the percentages yields between 5 different lines. Obviously, this tells you even with variability, you are achieving high numbers for these markers for RGCs. And these are the different stains we use to characterize them. Here in this slide, this is the gamma synuclein derived GFP expressions. So these RGCs are labeled with AAV2, adenovirus 2 cassette, which drives gamma synuclein expression, and these are labeled green. So these are the cell bodies, and then these are the cell bodies, and these are the connections they make. So this is day 45, and you can really appreciate how they actually look in the dish. These are the other markers which they stay positive for. And we also looked at, so I showed you that they're 90% pure, and then we tried to look for what other cells are out there. And the only thing we could actually trace or look for is the astrocytes. We found at least 4% of them staying positive with GFAPs, and they're making astrocytes. I don't know how we end up with 4% over there, because this we have checked. We do find them. What we found was if we start with a lower percentage or lower pluripotent cells, like 85% or 80%, we do see these numbers go up. We do see these numbers go up to like up to 10%. So that's why I said at the very beginning that the pluripotency is really critical to minimize any other cells in the region of protocol. We also record it from that. So what we do is just like any neuron responds to the stimulus, electrical impulses that we give them, and then when you actually tease a neuron, when you give them an electric shock, let me say you give them electrical impulse, and then they respond, and then you can actually record these stimuli they get. So we have to test them, or we have to record from these to be able to say that these are RGCs and not any other cells. So these cells are capable of doing that as well. So we also did single cell sequencing, single cell RNA sequencing. I'm sure most of you are familiar with this. This is you take these cells, all of them, and try to make several libraries from each one of these cells. And then when we do single cell sequencing, isolate them into clusters. The reason we did this is to see how many kinds of RGCs that we see in our differentiation. And then we identified more than 25 types of RGCs that we found out. So this is manuscript is currently under review. What we are doing now is once we confirm the subtypes of RGCs, we have these molecular markers now. We have to go behind the rig and then kind of record from each one of them to really make sure, establish that these are different kinds of RGCs, and then that's something we're working on right now. Coming to the part that you guys don't like much is, so now what I've shown so far is we took some iPSCs, we were able to differentiate them into RGCs. But what next? How do you actually scale it up? How do you actually make it? How do you help the patients? Obviously, you can actually study the cells, understanding pathways. I've actually moved a couple of slides here where we standardized different chemicals and saw different pathways that go up with oxidative stress, NMDA treatments, and all the other things. We did everything we could do that what to be done in a dish to really establish that these are RGCs. They can be used by anybody to not only study axonogenesis, for example. We are collaborating with Ohio University, and they have some growth factors we are working with, which increases the axonogenesis, which promotes axonogenesis by 400%. So that was a project which we are still working on, but those initial results really blew us away. So what I'm trying to get at is we can use these cells as model systems in a dish, and we can interrogate all these questions we have. And that's most of the aims that we kind of explain to me about the center here. But we wanted to go a step further and see whether these RGCs that we make can really make connections. Because at the end of the day, if you make these cells and that you're saying these are RGCs, if they're not, if you're not going from bench to bedside, how impactful is it? At least for the US, they would want a human connection for disease and really make it work. So what we did was we tried attempting transplanting these in the eye and then seeing if they make connections, because that's the next step. Because I've shown you a 9-layer complex retina wherein the RGCs are the last layer. If you get signal from the photoreceptor outer segments, if the RGCs are actually not making the connection, no point restoring whatever you have in there. But if you're not receiving the impulse, if the connections are not made with the bipolar cells or other cells, you can't take the signal to the brain and the person still can't see. So that's what we wanted to establish. So we took the donor iPSCs, we differentiated them into day-36, they found these nice bundles. We found, for the purpose of identification in an eye, what we did was we transfused them with the AAV2. I said, this is a vector which is actually patented to study the RGCs. It has a GFP driven by a gamma cyclin promoter, which is patented to supporter. They label the cells green in as less than 3 days of transduction. So around day 40, RGCs, you actually purify them, you inject anywhere from 50,000 cells to 1 million cells. Say inject in 2 microcellular, and then we chose mouse retina just because to keep our budgets to a minimum. We would want to go higher. We are in talks to try this in monkeys in another 4 months. But at least we wanted to show that establish a proof of concept to see if the cells work, integrate, and then we'll see. So we did these intravitreal injections in the mouse retina, and then we did a non-invasive observation and an invasive observation to assess the positiveness of our experiments. We'll start. Why is it not working? So quickly, these are the fundus images captured at nerve fiber layer. We use blue light effect and scanning laser ophthalmoscope. So basically, you're looking into an eye with a laser, with a light in front of you, looking into the eye, and then trying to make sense of what you see there. Right? So a whole group. Imagine a doctor looking into your eye and then seeing this in the very back of the eye, in the nerve fiber layer, like in the different eye in the nerve fiber layer. So these mice are the mice we followed up 2 weeks, 4 weeks, 6 weeks with a injection of PBS. It's like a control injection, and then we wanted to see if something happens here. And we also use micron-P, which actually captures the autofluorescence coming from the blue-green spectrum. So normal life mice look okay. We looked at up to 6 weeks, they look okay. There's X here, but that's fine, those are normal. And then we looked at OCT, which tells you this is the thing, this is the retina that you can see. This is a non-invasive procedure. Shine light and measure the depth of the retina. Look at all the individual layers. He's an expert in actually doing that. So this is the mice where we actually gave the injections with these treated cells. And then when you follow up, this is 2 weeks post-injection, 4 weeks, 6 weeks. And then this is the color image. What you see here, the green cells, which we are labeled prior to transplantation, and they spread. These are the intraretinal injections. There is no focal distribution of it, but you want to do the sporadic distribution across the eye. And what we are learning now is the pattern that you see here. This is the OCT, where you have, you do see some noise here. This may be partly due to the inflammation, or this may be partly due to the iPSCs, which haven't integrated yet. So we're still doing more studies on that, but we haven't observed major inflammation or something after 6 months. So they can be either cells which are waiting to kind of go and integrate, because we chose a mice which is 4 months old. So they are quite old enough. They don't have any spaces in the retina for them to go, for them to accept the incoming RGCs. So the debris usually settles down by 6 weeks. I wanted to show you how variable they are, because I just showed you one mice over there. So you can appreciate that these are pretty consistent among all the injections we did. And so we have to do both male mice and female mice just to make sure that we are not biased. Right? So this is a part of any experimental design that we have to do collaborated with VESIT. We took Brent Bell's he's an expert in actually analyzing these fundus images. And what we did was after feeling confident that now we see something in there, we wanted to see what's in there. And we went after 6 weeks, we actually euthanized the mice, and then we wanted to see what are we seeing. We cut these sections of the retina, and then we found out that these green dots that you see here are actually the ganglion cells, and they're exactly integrated in the site where we wanted to integrate. We do see 1 or 2 nuclei in the INL layer, intranuclear layer, but that's pretty common. It is known that they are there, and we actually extend projections for this side. But we see there's a few of these and many more of these, and this is a pretty usual occurrence, especially when you are doing cell-based therapies in intravitreal, and you have to really tune our methodology to increase more of these and minimize these. When we did a flat mount, what's a flat mount? So you take the eye and then you actually lay it flat. Because here what we did was we sectioned the eye and then we see the dots. But if you want to really appreciate whether they're integrated, whether they're branched out, what we do is we actually lay it out flat and then we capture these images using a 2-photon confocal microscopy. So you see these are the cells, these are the transplanted human cells, which not only integrate, but you see, look at these architecture, look at these branching that they do. They're beautifully integrated and then split the beats. And they actually also need bundles. So we really got excited, and then we thought, okay, what are these stacks of each of these images that we see, and then we try to generate, we did 3D rendering. What you can see in this video is they not only are in one plane, they can actually extend beyond the planes, and they're trying to make these connections. So then, okay, so now we know they went in there, they integrated, they made these connections. So now we wanted to see how they function. The only way we can assess them is to record from the a very classic experiment that we did with normal cell stimulation. So you do see that, these actually increase firing. So here, this is the place where we actually, so these are the cells that we injected, and then this is the red dye we use before, like patch clamp. It's called, I think it's called patch clamping and whole cell clamping. So you inject the red dye to make sure that the cell that you're studying is the cell you're recording. So you inject the red dye in there, and then you polarize the yellow, you know before and after the experiment that you wanted to record from one cell and you indeed recorded from that cell and the response that you see specific to that cell. And so you do see that there is increased firing of response in light-induced polarization. So these are very time-consuming experiments. If a person sits, so Sergey is the architect behind these experiments. So in a day, if he sits for in 12 hours, he can record from 6 cells. Patch and record from 6 cells. It's a very time-consuming process, but we were able to show at least there were like 5 light responsive RGCs and then 1 not light responsive. One is an off cell, the other is an on cell. So these were more experiments to further show that. And we also injected, so I said, in research, we do many different ways. So then we need to see whether they stain for all the RGC markers. And then they need to stain after integration for all the RGC markers. One question you may always have is, these are human cells integrating into the mouse retina. So if I show you all these pretty pictures and tell you, ask you to believe that these are the transplanted cells, you wouldn't believe until I stain specifically for a human stain using a human nuclear antigen and say, well, whatever the green you're seeing here actually is a human nuclear antigen stain cell. And when you see a merge, you see a yellow here. Then you're confirming that the cells that I'm showing you here, the cells that we characterize here, are indeed the human cells and not the mouse cells, which actually also accepted these markers. So these are very few sections. We're still actually preparing a manuscript and we are adding more data to this. What this shows is, so we were able to establish that they integrate, but the challenge in the field right now, in the field of glaucoma, is, well, you make these beautiful cells, and you showed that the cells integrate. Can they go to the brain? Because if they don't go to the brain, if the axons don't extend to the brain, the mice can't see, the patients can't see, nobody can see. So that's the next challenge. And there are a lot of researchers working on different kinds of experiments, like deletion of genes like P10, which will actually increase the axons, not axonogenesis. So we wanted to show, or we wanted to see these actually extend axons and go into the optic, going to the optic nerve. So we cut the optic nerve and then so these are very minute sections, 3 micron thin sections, and you cannot get any better than something what you're seeing here. Whereas if you are actually seeing a green stain, the green here indicates the cells that were coming out of the transplanted RGCs from human origin. And this is the neurofilament which changes the neurons. And then what you see here is an overlap, which is yellow, which says that these are not only RGCs which are trying to go back, but they are in optic nerves. Conclusions, I'm not going with that, but like I think I did summarize that. Okay. We did see the subsequencing, we have established this protocol, definitely we generate from any IPSCs to take in, we definitely will get 85% positive, that we get more than 80% pure IPSC RGCs, and you can study pathological mechanisms like oxidative stress. We have already done that, I haven't shown much data here. And transplantation, and there was one more researcher who published their work from University of Pittsburgh, who tried this methodology by isolating RGCs from a mouse retina, and they have showed that they can integrate, they have success of 65%. But this is mouse to mouse. This is mouse to mouse transplantation. Our methodology is reduce 85%. There's no issues with the species here in the mouse, and the mouse seems to like them somehow. But obviously, we want to go higher. But this was just a testing model that we wanted to actually do. At the end of it, this is something that we are need to work on. We help project and everybody who's interested in working on this to increase, improve the delivery, maximize the numbers that we integrate, and also maybe do a dual combination of packaging some small neuroprotective compounds, and and then held the RGCs that are already in the vitreous till they hone into the RGC. So this is an area still under development, and we want to maximize this. But this is still the best when compared to what is reported in the literature. We have documented over 100 or 1,000 different RGCs that are integrated by counting manually or flat amount. We have done experiments in close to like 12 mice so far. We have more mice. We are still analyzing them. What else? Yeah, this is what I actually explained this to you again. So I want to, get you back and get your attention here. I want to show these are the cells that you see them. These are dynamic videos. These are taken from day 25 to 35. The video is granular, but like what they do is these RGCs, they actually really form nice, beautiful connections. And each cell, if it's actually labeled GFP positive, they try to come together. All these clusters were formed by individual cells coming together and actually making these bundles. This is in the dish model. And let me show you another video. So this is another video where we try to show, I've actually shown you the 3D rendering, but this is in the mouse retina. You can appreciate how these are making connections, and you can see all these connections between these cells. And we actually dive in using you can really appreciate all the connections they are making. Really telling that these not only stay there, but they integrate, they actually are really happy there, and then they're extending their neurons. And as we showed, they're actually knocking out as well. We are publishing this work. Maybe I will be submitting the manuscript next week or so. My trip to India definitely delayed it. But these are the future directions we are trying to work on and improve. We definitely want to improve the transplantation methodology, I said, using some growth factors to work to, like that. And this is something that we have been working, and it's really good. So so far I have showed you that this is a control mice I took, and I injected them, and I showed that these cells can really go and integrate and work. Now coming to the disease model, just like working in a glaucoma person, where you have less RGCs. How do you actually get less RGCs in a mice model? Because what I think is if we kind of create some room for the incoming RGCs, there is better chance of more integration or even better integration. So I think using NMDA, we have standardized the dose of NMDA, which is actually a neurotoxin in a way. It kills the RGCs. We have standardized the concentration at which 50% of RGCs get knocked away within 2 days of injections. And we are transplanting, we are taking those mice and we are transplanting them. Even since our experiments were done like within the last 6 months or so, we did not have many mice. But I'm happy to report that in 6 mice, out of the 6 mice, in 2 mice, we did a functional recording. So in mice, just like humans we are able to see and communicate that we can see. In mice can communicate, but in their own way. So we put a drum around them, we put lights in the drum, and then we shine the lights in. It's a 360-degree drum. We make the mice sit in one place, and then kind of see where the light is. The mice definitely does turn their head to see where the light is. So when we did an NMDA injection, when we transplanted them with RGCs, 50% of the mice that we injected them have regained function. Although it's very preliminary, but I'm still very happy about the result we got because it's really positive. And I'm sure by doing more numbers and then optimizing our delivery and everything, I can increase those numbers. But that's just a start. Oh, this is my team. I couldn't do any of this work without the support of my chair and the wonderful team I have. I have a small team here. The team, these 2 people, Matthew Hyder, went to the company after learning all the IPS work. It's brain, obviously. And Sonia, she is in Germany now. She played a big role in actually initial standardizing methodology. This is my small team. and I'm still looking for positions if anybody is interested to apply. So I got the funding from Right Focus Foundation and Research University Foundation, who sponsored most of the stem cell work we did. Other areas of research, just because I taught glaucoma, that's not what we do. We are also looking into microtherapy. We did look at, I've shown you that we were successful injecting just the RGCs that we made, but we're also transplanting mitochondria, and then that project is equally successful. We have some preliminary results from there. We're trying to elect the funders now because we need more money to study that and bigger team to study that. So this is something we're doing. This is a big part of our research too, which I did not talk here. We do a lot of A&D research too. So A&D research, as a lot of my postdoc topic is actually on A&D. And my specific interest in AMD is the lipid biogenesis pathways in AMD. As you know, in AMD, glucose play a big role. And my interest was to explore the lipidomics, or the lipid pathways that are responsible for forming glucine or gluconeogenesis. What we do as with iPS-RTCs, we take iPSCs and differentiate them into epithelial cells, retinal pigment epithelial cells, in about 35 days. Same time, around the same time, we make these beautiful RTEs that people are actually now using for transplantations as waves now, with the help of a desk surgeon here. It's very possible to do these over here. And my interest was, as I'm a functional geneticist, so I'm studying the role of two genes which have been associated with AMD. One is the hepatic C, and then phosphorescent protein. We did a lot of EQPL studies. These are the expression quantitative trait loci. We are assessing. There are a lot of SNPs. For example, I don't know if you heard more about GWAS studies or not. So every now and then you see a Nature paper saying, okay, we found this gene in Indians or maybe South Indians, and then this gene is responsible, is associated with one particular disease. And they'll say a couple of SNPs are responsible for that. But maybe not all the SNPs that they found are the true causative changes to gene expression. So what we do is we take a step ahead, and this is something we definitely do here, is we do EQPL studies. We take out all the SNPs and then we assess the gene expression. And we do in tissues, we use tissues for that, we do RNA-seq, and we do an attack seq, and then we combine the data to really look into, we can even look at the 4C content, 4C capture, you might have heard about 3C, 3D chromatin capture. You have to use these techniques to really understand how a SNP is associated with the disease and how it's actually modifying an expression of the gene, either cis or somewhere in the genome that trans. So we are doing that currently. We have really gotten much better at doing CRISPR-Cas9 knockouts, although CRISPR-Cas9 is very widely used. I'm sure you guys are doing it too. We've gotten much better at it, and we can knock out any gene in a span of 40 days, screen the common population, and we have our hits. So if you're interested in a gene, we can knock it out in 40 days in any cell type of your choice, and especially iPSCs. And we also are interested in, so this is a collaboration from the UK that I have. I got interested in the methylation, how it affects aging, how aging affects methylation of some of the genes. In AMD, this is very less explored, and in glaucoma, it's not explored at all. So those are two avenues that I'm really interested to work on. And that's pretty much it. You guys have any questions? I know maybe some of it went over the top, but I can try to explain as much as possible. Even online, I don't know if they can ask any questions. If anybody's there, please, go ahead. Go ahead.
So, use IPSCs, what is the units of basically that?
These are human, these are Caucasians. Caucasians thoroughly screening their IPS populations.
The constraints that were used for studies.
No, these are all the control lines. These are all the control lines with rich clinical data. I would say, so these are less than 40-year-old patients with no known history of any eye disease so far. They might have blood pressure, those comorbidities after 40. But right now they are controls. They still continue to be controls in their current system. So far, I know in the last 10 years. So, but these are not commercially available like to develop these types.
I don't know if testing towards
Which one?
this
Mesenchymal stem cells. Mesenchymal stem cells, no, we haven't tried them. We wanted to use them as delivery vehicles for mitochondria, but we haven't looked into that. Yeah. But that's a good solution.
because I think this is not very hard to work back. They are
Working that
more vulnerable to getting testing a lot of other issues in a big niche. Whereas FSCs are like more destined towards they are like more controlled on the pills. So doing a number the FSCs would have been, do you think it would have increased the efficiency of integration to a greater extent?
I haven't thought about it in that way. I was more confident about how successful we got with the IPSCs. They haven't gone back. Once they are RGCs, they remained RGCs.
Yeah, because there's a very high probability of IPSCs working back. IPSCs post integration.
But even after differentiation,
Even after differentiation, because they are more, I would say they are more rowdy cell lines. MSCs are like a little more on the evolutionary line and creating a stable knockdown MSCs. Testing was formation
that's a great
channel.
suggestion.
The probability of integration and the probability of PTC integration are very pretty much higher. You use.
I'm definitely going to do that. That's a good suggestion.
Yeah, I was just wondering what is the logic here why you went for an IPSC?
Oh, IPSCs. IPSCs are more easy to, okay, the goal of actually going with IPSCs is actually making a patient specific therapy. So anyway, I would like to use this technology to actually get a line.
And more personalized.
It's more personalized medicine because it's instantaneous. You don't take a couple of years to develop it, and it's like that. And then you kind of have to go with immune rejection, like tackle immune rejection at some point. Even though we don't much here, we look at some immune related markers. So that's the easy basis.
Not much concern. Limitations of the immune cells present in the eye.
Yeah, the eye.
It's because it's a very closed system. The probability of immune cells really getting into the eye. So the probability of rejection is very less, minimum as compared to people.
Yes, I agree with you on that.
So I think that is why I was saying that MSCs would have been a better option, a better choice of system.
It's more a personalized medicine option that we initially went with, but definitely I'll look at that. Any other questions you have? Any other questions you have? Yes. Potency for what?
IPSC.
Yes. So are you talking about pluripotency? Oh, we have markers, and we use fact sorting, and we use markers, 3 different markers, and then we check, and then we average the readings we get from all 3. We try to say more than 95%, but usually we end up close to 90%. That's the maximum we end up at. Go back to these. Any other questions?
So, on behalf of LSP and on the party on behalf of Untitled Chapter, I'm really thankful to Dr. Murthy to really do this precious time, this short trip in India, and at least give insight to students on what are the opportunities that are there for us, even though we are not aware of the kind of global research that is happening with a small issue like I, which has a lot of significance. I think 99% of the world is focusing on cancer and it's focusing on diabetes. But there are the statistics that Sir has shown in the first slide. There's a huge population in India itself which is suffering from this not so lethal, but yes, a concerned disease, which is partial blindness at a very young age because of bad lifestyle or your probability of getting into diabetes because of genetic inheritance. So these are very, very niche areas of concern and they require very high level expertise, whether it is research or whether it's therapy or a humanized approach to the treatment of the disease. And I think what Sir and his work has given us an insight of that even though the eye may look so small, the nitty-gritties of it, doing its function is so complex and it's so integrated and it's so well coordinated by nature that even a small neuronal imbalance can actually cause very severe implication or you not being able to see or having blurred vision. So these were not my concerns as a researcher because we were looking at very big diseases like cancer and tuberculosis and HIV, which were of greater concerns. But I think the kind of research that we have shown, it has really opened up the next level of enthusiasm.
Yeah, I'm glad actually I introduced you to the topic.
Yeah.
But you have a well-renowned expert, I would say, in front of you, who's working in eye research, eye research as well. And he's like the god for surgeries. I don't know if he, I'm sure you know about him, but my interpretation of him is he's the god for retinal surgery in India and across and the entire world. I want to take a minute of this time to explain what he does, and I want you to appreciate and participate in what he does, because obviously this research cannot move ahead without the collaborative efforts, without the teamwork, without the collaborative projects. And I came here. There are many different reasons why I came here. Definitely looking into opportunities to see, well, can I come back? That was the first question. And second thing is, we've been talking about collaborative opportunities with Aditya Jyot, and then seeing what kind of research that we want to do over here. And I'm sure you know, cataract surgery is a very common surgery. I'm sure each person in our family, extended relatives might have gotten it, right? In 15 minutes, right? And the retinal surgery can take anywhere between 1.5 hours or 1 hour 15 minutes to 1.5 hours. And the God sitting in front of you can do it in 15 to 20 minutes. So that actually an achievement. I'm sure he has gotten all the national laurels for the same reason. So he's so energetic. He's a postdoc. He's a postdoc in the first year of my after my PhD, and so energetic, so vibrant, so connected. I'm sure you have a wonderful, so passionate about his work. I'm sure you have wonderful opportunity outside your work here to go personally engage, learn more, contribute to this research, and really take this the next step. Because I think that's the message here. The message is to see what we are doing over there. All this has been done in 1.5 years, 2 years during the pandemic. Thanks to all my team, they have worked in the pandemic, looking at these mice, turning up every day, wearing the masks on, with all the stress. But coming back here, doing similar collaborative work, it cannot be just done by one person. He can motivate you, he can guide you, he's a mentor, collaborate, everything. But we need you and your friends and anybody who wants to get involved. Just learn. I mean, obviously you can open a book and start reading about the eye, and you can get hooked like that because it's a window to the brain. He actually can look into the eye and tell you, maybe you have an issue. I think you are diabetic. He can look into the eye and he can tell you if a person is diabetic, right? So it's a window to the brain, and he's not looking in the brain, but he's working in the eye. But it's amazing, So I hope you could learn something from what we presented, and there's more in one of the research topics to work on. but we have a much bigger role to play now. Now that you learned about it, in another opportunity, if you give me another opportunity, I can talk about the AMI work that we did, and equally enlightening to what else can be done over there. But more needs to be done. Keep pushing, and keep learning, and keep reaching out, and you're always there to help.
Thank you so much for offering that. And I think this is an opportunity to practice and take up one of these projects once further in research, because they're young budding scientists getting into their research world, and if any of these topics or any of this related is fascinating. So I'm there. To make sure that.
And then right in your backyard. Right in your backyard. I just walked from there. I physically want to walk to see how far the place is.
In the neighbors, so.
And the Institute of Chemical Technologies again.
Taking a lot of compounding can grow at these cells. You can do so many things, so many wonders. And then maybe eventually you can go to the brain. So you can do neurons too. So sorry, I do
This is on behalf of the
Thanks a lot. Thank you.
Thank you.
Thank you for the opportunity. Yeah.
I think I hope to increase the interest for all of them.
Sure, please.
You don't have any other questions, do you? Go ahead.
Thank you all. Thank you all. So this will be available in the YouTube. And thanks to Ritesh, thanks to Ronali, and our